My name is Marcus Hafner, and I am a tenure track at NIAMS, and it is my pleasure today to introduce Dr. Dinshaw Patel, who visits us from New York, where he is the Abby Rockefeller Mosee Chair in Experimental Therapeutics and a member in the Structural Biology program of the Memorial Sloan Kettering Cancer Center. He is one of the true titans in the field of structural biology, and his long and distinguished career revolves most part around the biophysical dissection of nucleic acid structures and nucleic acid-protein interactions. I got to know Dinshaw's energy and commitment to excellence firsthand in 2007 during my postdoc with Tom Tuschl, who was his collaborator on a fantastic string of papers that for the first time reported on structures of full-length Argonaut proteins bound to their RNA targets. And these proteins are at the core of the RNA interference pathways. So Dinshaw has been studying this pathway since its emergence in the early 2000s, and his elegant crystals of RNA components from multiple organisms greatly contributed to our current understanding of small RNA pathways. He is a pioneer in numerous other fields as well. He contributed the first structures of multiple ribozymes and metabolite-sensing RNAs, so-called riboswitches. He provided the structural basis for epigenetic regulation by histone marks, as well as DNA damage resolution by bypass polymerases. He was involved in the dissection of a novel innate immune pathway sensing DNA, the CGAS-STING axis. And finally, he is also elucidating the structural basis of CRISPR-Cas systems. A few words about his career. Dinshaw is a native of Mumbai in India, where he also attended college and obtained his bachelor's in chemistry. He then moved to the US as a Tata Fellow and earned a master's degree from Caltech, followed by a PhD from New York University. He stayed in the area and first did a postdoc at the NYU and then moved to the legendary Bell Labs in New Jersey, where he became a member of the staff in 1970. In 1984, he was appointed professor of biochemistry and biophysics at Columbia. And finally, since 1992, he's a member of the structural biology program at MSKCC. Of course, he's the recipient of numerous awards. He was a distinguished staff member of Bell Labs and a distinguished alum of NYU, the president of the Harvey Society. He is the recipient of the NIH Director's Transformative R01. And finally, he is a member of the American Association of Arts and Sciences, as well as of the National Academy of Sciences. Today, Dinshaw will give us an overview of multiple aspects of structural biology of RNA-mediated gene regulation. So please join me in welcoming Dr. Dinshaw Patel for this exciting lecture.
Thank you very much, Marcus. My talk today will focus on our work centered on RNA, its recognition, its catalytic activities, and I'll do that by describing work on riboswitches, mainly focused on RNA architecture and the generation of pockets that recognize small metabolites. I'll talk about ribozymes, self-cleaving ribozymes that can cut their own RNA. And finally, I'll talk about very recent work on CRISPR-Cas systems. So let me start with a brief background on riboswitches. These are metabolite-sensing non-coding RNAs, shown schematically here in two forms. There is a sensing domain and there is an expression platform. Metabolites are recognized by the sensing domain, which undergoes a conformational change, which is then propagated to the adjacent expression platform. So that some marker in the expression platform, let's say, is exposed in one state, and on metabolite binding gets sequestered. So depending on the concentration of the metabolite, you have an on-off switch. To show you an example, Here is a riboswitch for the metabolite thymine pyrophosphate. Here is its sensing domain, and here is its expression platform. In the absence of metabolite, you have these pairing alignments shown here. When the metabolite binds, you have an alternate pairing alignment, so that in the free state, the Shine-Delgado element is exposed. In the bound state, it is sequestered. So translation occurs in the unbound state, and it's inhibited in the ligand-bound state. We would like to study the structure of these riboswitches as well as their ligand recognition pockets. Generally, this is feasible for the sensing domain because when the ligand binds, the structure becomes compact and can be studied crystallographically. So the emphasis is on the overall RNA architecture, the binding pockets, the ligand recognition and discrimination, and some of the transitions that I mentioned as you go from the free to the bound state. So let me just show you the structure of the thymine pyrophosphate riboswitch bound to the sensing domain. It contains a three-way junction which, when thymine pyrophosphate binds, adopts this tuning fork-like architecture, where there's a stem, a handle, and two prongs. You have a hairpin loop here interacting with its receptor, holding these two prongs in a parallel alignment. The three-way junction is here, and very interestingly, the ligand, which contains a heterocycle at one end and a pyrophosphate at the other, binds above the junction in an extended form where there are pockets in both helices. In one helix, you recognize the heterocycle, and in the other helix, you recognize the pyrophosphate. So this was very interesting. How does an RNA, which is a polyphosphate, recognize a ligand which itself has phosphates? So if you blow up the binding pocket, remember there's one pocket in this helix and another pocket in this. This is what you see. Here is the thymine pyrophosphate. The heterocycle sits in one pocket where it's sandwiched between purines, and its polar edges are hydrogen bonded. So this end is anchored. And at the other end, where you have the pyrophosphate, the phosphates are coordinated to hydrated magnesium ions directly, and then one of the magnesium ions is in turn coordinated to the RNA, not to the phosphates of the RNA, but to the base edges of the RNA. So RNA can fold, generate a pair of pockets, anchor the heterocyclic ring, and accommodate a biphosphate by using magnesium ions, hydrated magnesiums, as bridges between the ligand and the RNA itself. So this was quite novel, the dual pockets, the extended structure of the ligand. And what we don't know is what the conformation is in the free state, whether these helices are parallel or they are fraying, and only in the presence of the ligand are they brought together and aligned in parallel. So what about pockets? How small a pocket can one generate using an RNA backbone? So here I turn to another riboswitch. All these riboswitches were identified in the lab of Ron Breaker at Yale. And this is a riboswitch called the fluoride riboswitch, which is involved in activating the expression of genes that encode putative fluoride transporters. And here is the sensing domain of this riboswitch. It's simple. It has a stem, an interval bubble, and a stem loop, and some dangling ends. But this riboswitch binds fluoride anion, which is, of course, extremely small and also negatively charged. So how does RNA form a very small pocket and accommodate a negatively charged ion? So Amy Wren in the lab, when she solved this structure, the sensing domain of the fluoride riboswitch, bound to fluoride anion, this is the overall topology. The Breaker lab that identified this riboswitch realized that this segment here could pair with this loop segment to form a pseudoknot. And indeed, in our structure, you can see the pseudoknot forming a duplex in purple. And the various elements are color-coded here and in the overall structure. And what we find is that the fluoride anion is positioned in the center of this three-dimensional architecture. So how is it accommodated in its pocket? And here we see something quite remarkable. Here is the fluoride anion. It's coordinated, which is negatively charged, which is coordinated to 3 magnesium ions. And then the magnesium ions in turn are coordinated to phosphate oxygens and water molecules, with the phosphate oxygens directed into the interior of the RNA scaffold. So you have a fluoride anion surrounded by 3 magnesium ions, and the magnesiums in turn are coordinated to phosphate oxygens and to water molecules. So a negative charge surrounded by positive charges, surrounded by negative charges of the phosphate oxygens. And the whole concept is that fluoride anion is very small. You cannot form a small pocket, so you enlarge the pocket by not only having fluoride, but 3 divalent ions. So the pocket becomes much larger and now can accommodate the fluoride anion coordinated to the magnesiums. Now this result was quite striking and we were concerned, so we looked at the protein world for proteins that bound fluoride anions. And we found an example shown here, which is compared with what I just talked about in the RNA world. So in the RNA world, you have a fluoride surrounded by 3 divalent ions, surrounded by phosphate oxygens. In the protein world, we have a fluoride anion for this example, surrounded by 2 divalent and 1 monovalent ion, and the phosphates are replaced by carboxylates. So nature has used similar principles to recognize fluoride anion by using divalent ions and inwardly pointing either phosphate or carboxylate groups. Now, we're very interested in the conformational transition as you go from the ligand-free to the ligand-bound form. So here I want to show you an example where we could monitor the sensing domain both in its ligand-free and its ligand-bound forms. And this is a riboswitch that binds the amino acid L-glutamine, and the riboswitch is positioned upstream of genes involved in nitrogen metabolism. Again, the topology of the sensing domain is very simple. It contains 3 stems and an internal bubble. Now, to solve this structure, we had to add a U1A RNA loop, bind it to the U1A protein, and solve the crystallographic phase problem. But for this sensing domain that binds L-glutamine, we were able to solve the structure in the free form. So you have 3 stems, and once again you have that tuning fork architecture, a handle, and 2 prongs. And when we look at the entire scaffold, there is a GGA element which is disordered in the free form. One can ask what stabilizes this free-form structure in the absence of ligand, and what we find at the 3-way junction are a set of stacked bases stacking on their neighbors. When we add L-glutamine, the conformation changes dramatically from the tuning fork architecture to an L-shaped architecture. And at the junction of the L architecture is where the L-glutamine binds. Further now, the segment that was disordered in the free state, GGA, can be followed in the bound state, G22, G23A24, forming one side of a pocket where the L-glutamine binds. In addition, we form a long-range interaction between this G and that's C to form a Watson-Crick GC pair. It's an open pocket in which the L-glutamine binds, and again the pocket is enlarged by a hydrated magnesium ion. So it's an open pocket, in part accounting for the weak binding affinity. Here is L-glutamine sitting in this pocket together with the hydrated magnesium ion and the long-range GC pair. And what you observed is all the heteroatoms in L-glutamine are involved in either direct or water-mediated hydrogen bonds. And this is important because this riboswitch binds L-glutamine and discriminates against all other amino acids, including their D analogs. So what we have observed for this riboswitch for L-glutamine is that we are going from a conformation in orange in the free state with a disordered segment to a conformation in blue in the bound state, a large conformational change, with formation of this long-range tertiary pair with the binding pocket located adjacent to it. Now, we are confident about the conformation that we see in the crystal for the glutamine bound form, but there are questions about the glutamine free form. Is it the conformation we see in the crystal, or are the P2 and P3 helices adopting or sampling different conformations? So we worked with the lab of Hashim al-Hashimi at Duke University to use NMR, both chemical shifts and residual dipole couplings, and also molecular dynamics simulations. And to summarize that result there, we can explain the residual dipolar couplings very well for the bound form, but the indications for the free form is that the free form is sampling a range of conformations. And in this range, you either have a tuning fork or an L shape, but you don't form that long-range tertiary pair in the presence of magnesium. Only when you add L-glutamine does it trap the bound state, which forms this long-range pair. So the free state is sampling various conformations. In the presence of magnesium, that equilibrium shifts, but you require glutamine to lock in the bound state conformation. The last example I want to show you is to address the following point. You have an RNA scaffold, you have a natural metabolite that binds somewhere in the scaffold. Can we use selection methods to identify small ligands that could also target this particular pocket? So many years ago, we looked at this six-stem junction RNA that bound the metabolite flavin mononucleotide and solved its structure. And interestingly, despite no indication of pseudo-twofold symmetry, you have one half of the RNA related by symmetry to the other half, and the FMN sitting in a junctional pocket itself in an asymmetric orientation relative to these two symmetry elements. Now, many years later, a group at Merck identified through selection a small molecule which they called ribosyl, whose structure is shown here, and it's a highly selective modulator of bacterial riboflavin riboswitches by repressing riboswitch-mediated gene expression and inhibiting bacterial cell growth. So here is the FMN sitting in its pocket. It's sandwiched between two purines. Its side chain has a phosphate and a hydrated magnesium ion. And here is ribosyl sitting in the same pocket using these two ligands, which are planar and sandwiched between two purines, and this moiety then allowing stacking of this heterocyclic ring with a guanine. So these pockets can be targeted by other man-made ligands, in this case repressing riboswitch-mediated gene expression and inhibiting bacterial cell growth. Now I want to move on to the second topic, which involves self-cleaving ribozymes. In the literature, for many years, there have been self-cleaving ribozymes with names like hammerhead, hairpin, Warcourt satellite. And they involve in rolling circle-based replication of satellite RNAs. And for a long time, no new self-cleaving ribozymes were identified until again the Ronald Breaker lab found a set of ribozymes, small ribozymes, with names like pistil, twister, and hatchet. And for each of these ribozymes, there was a particular position in the backbone where cleavage occurred. For pistil here, for twister here, and for hatchet here. Now the concept is, why does cleavage occur at a particular site? And based on the earlier work, if you have a dinucleotide step, what happens in nucleolytic cleavage is that the 2' hydroxyl at this position is somehow activated. It targets the PO bond. It needs inline alignment to do that. And you get PO bond cleavage. So you need something to activate the 2' hydroxyl. You need something to stabilize a negative charge that's developing at the cleavage site. And the concept is there's generally a general base that does the activation and a general acid involved in the stabilization, and one needs inline alignment. So the important points are a specific cleavage site, inline alignment, what are the catalytic residues, and is there a role for magnesium ion? So what I'm going to tell you today is the structure of the pistil ribozyme and a ribozyme called twister sister that's related to twister, and what can we learn from these structures. So here is the pistil ribozyme. It's a very small RNA. It contains a stem loop, these two stems, and an internal bubble. And from the sequence, the Breaker lab was able to show that it most likely forms a pseudoknot. So Aiming Ren in my lab solved the structure of the pistil ribozyme. The RNAs, you have two strands. There's one strand here, and then there's a second strand here. We work with a chemistry lab in Innsbruck, that of Ronald Mirkura, who can synthesize the RNA, as well as incorporate any analogs at whatever position we require. So here's the three-dimensional structure of the pistil ribozyme. You can walk through it. You start with this orange stem. Then you have a pseudoknot in magenta. Then you have the green stem. And the rest of it is then involved in a fold back. And at the cleavage site, U54, G53, the bases, instead of being stacked, are splayed apart. So what is occurring at the catalytic pocket? So first I show you the structure on the upper left. Here is G53, and here is U54. At this site, the cleavage site, the bases are splayed apart. But equally importantly, each of the bases is anchored in place through stacking interactions or intercalative alignments on either side. So not only are they splayed apart, but they're anchored in place. The next thing you want to ask are, what are the residues in the vicinity of the G53-U54 step? And what we find is that there is a guanine whose N1 is directed towards the 2' hydroxyl that has to be activated, and there is an adenine next to the PO bond that has to be broken. So do these bases play a catalytic role in activating the 2' hydroxyl and stabilizing the transition state, as well as after the PO bond is cleaved? So the most simple thing we can do is we can replace these nucleotides, G40 by an adenine and A32 by a cytosine. Now this is a two-stranded construct. One of the strands, the shortest strand, is a substrate strand. One adds the two strands, adds magnesium, and the substrate strand goes down, and as a result of cleavage, there are two new strands. So for the wild-type RNA, for the crystallography, we had to replace the 2' hydroxyl of G53 by a proton, a deoxy, but for the wild-type, which is all RNA, the substrate strand gets cleaved in the wild-type system. But when we make these mutations, G42A or A32C, there is no cleavage, telling us that these residues, G40 and A32, are important for cleavage. Now, if you look at A32, the entry position of the adenine and the 2' hydroxyl of the adenine may play a role in the catalysis. So if you replace the hydroxyl by a proton, make this a deoxy residue, and do the same cleavage analysis, cleavage occurs, but at a slower rate. As far as the entry position of the adenine is concerned, if you replace the nitrogen chemically by a carbon to make the C3DA analog, you completely lose cleavage, suggesting that the entry position has an important role in the catalysis and the 2' hydroxyl has a role that is less important. We can do one more experiment, and that is to try and measure the pKa's of these bases. Guanine has a pKa around 12. The RNA is not stable during a pH titration, so we were limited to trying and estimate the pKa of 832. And so the way this was done was to chemically incorporate a C13 label specifically at the 2 position of adenine, specifically for adenine 32, and then do an NMR titration. And the pKa that we observe is 4.7, which is 1 pH unit above the pKa of adenine in RNA. So in the context of the ribozyme, the pK of the adenine goes up by about 1 pH unit towards a neutral pH of 7. Finally, we were interested in the cleavage rate, and to do this, the Mikura lab generated a construct containing 2 strands, but in the substrate strand, they replaced an adenine by 2-aminopurine. So in the free RNA, you will get fluorescence from the 2-aminopurine, but if it's part of a duplex, that fluorescence is quenched. So when you add the 2 strands in the presence of magnesium, you generate the riboswitch, cleavage occurs, the strands break and are separated. So you start with a certain fluorescence level. When it forms the riboswitch and the 2-aminopurine is part of the stacking, the fluorescence gets quenched, and then over time, as the strands disperse, the fluorescence recovers, and we obtain a cleavage rate of about 1 per minute. Slow, but still measurable. Now, more recently, we've worked with a second ribozyme called twister-sister, again in collaboration with the Ronald Mirkura lab. And for this ribozyme, there is also a structure from the David Lilly lab that I will compare with our structure. So if you can see this, you have a stem, a bubble, a stem, 2 hairpin loops, a stem, and another hairpin loop. And the cleavage site occurs at a specific site in this ribozyme at this particular step. When the structure of this ribozyme was solved, its overall architecture is shown here. There is one continuous helix here and a second continuous helix here. And the cleavage site, once again, consists of 2 bases that are splayed apart. Now, in order to adopt this topology, you have long-range interactions within the RNA scaffold. And I'm just showing you one of them involving residues in this hairpin loop that interact with a base pair here to form a triple. G35 forms a triple with this base pair. And then there are two more interactions mediated by divalent ions with residues further away in the sequence. U36, the adjacent base, interacts with these two residues through coordination via hydrated magnesium ions. So the RNA folds through a series of long-range interactions to form this topology. I apologize that the slides are not very clear. What I'm trying to emphasize here is that in the RNA secondary structure, there are a large number of conserved residues, and these are circled. They're all over in the secondary structure, but they're brought into close proximity in the tertiary structure. So you can see, even though in the secondary structure they are far away, they're highlighted red, and they're brought into close proximity. Now you have a junction here linking this residue and that residue, and this residue and this residue. And at the junction, what we see is that these alignments are brought into close proximity and mediated by a hydrated magnesium ion. And sugar hydroxyls are hydrogen bonding with phosphates across from them on both sides. So magnesium is very important as part of the structural component holding this riboswitch together. So once again, I'm sorry you can't see these clearly, but the point I'm making here is that our lab studied the riboswitch shown on the top left, where we have a four-way junction, and David Lilly's lab in Dundee studied the same riboswitch but lacking this stem loop and therefore containing a three-way junction. In our case, at the cleavage site, you have the bases played apart, C62, A63 in yellow, while in the Lilly lab, with one less stem loop, these bases are stacked on each other. And as far as the details of the residues involved around the cleavage site, in our case, the step is C62A63. The phosphate group is coordinated by the N1 of a guanine, and through a hydrated magnesium, coordinated to the other phosphate through a water molecule. In addition, there's a second hydrated magnesium ion whose water molecule is close to the 2' hydroxyl that has to be activated. In this case, the hydroxyl is not inline relative to the PO bond, nor is it inline in the case of the David Lilly structure. What they do see, however, for this C54A55 step is also a hydrated magnesium whose water is close to the 2' hydroxyl. So two different sister twister ribozymes adopting global very similar folds, but in the catalytic pocket adopting distinct alignments. And what we have to remember is that these are really pre-catalytic conformations, and we don't know what the transition state looks like. So for instance, in our structure, the adenine is anchored in place, but the cytosine is directed outwards, so that this non-inline conformation could, as a result of a conformational change in the transition state, adopt an inline alignment. So what we would like to do now is to do something that the Ferre-Desmarais lab has done in the past, to make vanadate mimics of the transition state, and in those mimics see how these adjacent residues are aligned and whether we have inline alignment or not. And for our twister-sister ribozyme, we could again mutate residues. For instance, we could mutate C62 or adenine 63 at the cleavage site, C62 actually accelerates the cleavage, the base that's directed outward, when you replace it by an adenine. But A63, when it's replaced by a uracil, has no cleavage. G5, whose N1 is directed towards the phosphate, when replaced by an adenine, again no cleavage, and so on. Now I want to switch to the last portion of the talk, which deals with CRISPR-Cas systems. So this is a very hot area of science. When a phage invades the host, it inserts its double-stranded DNA into the host. That DNA is chopped up by nucleases, and a piece called a spacer is then inserted into the CRISPR host. And in the CRISPR locus, we have these spacers representing past invasions separated by these repeat elements. The CRISPR DNA, CRISPR-Cas locus DNA, is then transcribed into pre-CRISPR RNA, and that pre-CRISPR RNA is processed in turn into mature CRISPR RNAs that contain the spacer and the repeat. The mature CRISPR RNAs are then taken up into protein effector complexes, where the proteins are either single subunit Cas proteins or multi-subunit cascade proteins. When the same phage now invades the host again, it inserts its DNA, that DNA is chopped up, and if the sequence of DNA matches the spacer RNA, the effector protein then cuts both strands of the DNA. So from a structural biology perspective, the labs of Jennifer Doudna and Osamu Nureki in Japan have looked at the single subunit Cas proteins, in this case Cas9, and studied their complexes bound in the apo form, bound to CRISPR RNA, and then bound to double-stranded DNA. What's important about these Cas proteins is that they contain two nuclease domains, one of them called HNH and the other one called RUFC. So in the schematic, what you're seeing, the Cas protein containing a recognition lobe and a nuclease lobe, bound to a CRISPR RNA, and sometimes you need a transactivating RNA, shown in red here, and CRISPR with transactivating would form your binary complex, and then double-stranded DNA would come in. A segment of the DNA would pair with the CRISPR RNA to form this D loop, so the target strand pairs, the non-target strand is unpaired. And then the nuclease domains, the HNH and the RUFC, HNH would cut the non-target strand and RUFC would cut the target strand. Now you need to recognize foreign DNA from self-DNA, and foreign DNAs have a PAM segment that is recognized by the Cas proteins. What is important in these canonical Cas proteins is that you need two nucleases, one to cut the target strand and one to cut the non-target strand, and further, the cleavages are blunt end. So the structural biologists have solved the structure of Cas9 in the apo form together as a binary complex with the RNA, and then with ternary complexes initially containing the PAM duplex and the target strand, and in the long term actually D-loop structures. So the mechanism is as follows. You have apo-Cas9, the CRISPR RNA together with its transactivating RNA bind. Within the spacer segment, there's a seed element that is stacked. The DNA comes in. The DNA is screened for the PAM sequence to see if it's foreign or host. And if it is foreign, and if its sequence matches that of the CRISPR, or complementary to that of the CRISPR RNA, you start to form a heteroduplex between the CRISPR RNA and the DNA, and the beginning of a stem loop that is then expanded to finally form a catalytic competent state where the two strands are cut. And in all these steps, there are conformational transitions that for canonical Cas9 have been determined by structural biology. Now, we got into this area late, as I said, when a new family of Cas proteins were identified by Feng Zhang at the Broad and by Eugene Koonin, a bioinformatician at the NIH. And I'll start by talking about two of them, CPF1 and C2C1. Their domain architectures are shown here, and the striking feature of these new Cas proteins is they lack the HNH domain, but they retain the rough C domain. And further, they had a set of interesting properties that distinguish them from Cas proteins. So for instance, CPF1 requires CRISPR RNA but not transactivating RNA, making it a simpler system for genome editing. C2C1, however, it requires both CRISPR and transactivating RNA. I said that these two proteins also have another feature in the case of CPF1, that they not only cut double-stranded RNA, but they're also involved in the processing of precursor to mature CRISPR RNA, and that came from Emmanuel Charpentier's lab. Once again, these two proteins have a single rough C domain. They lack the HNH domain, but yet they have to cut both strands, the target strand and a non-target strand. While Cas9 results in blunt end cleavage, these two proteins result in staggered cleavage of the double-stranded DNA. And finally, the PAM sequences are different. In canonical Cas9, they're G-rich, and they're proximal to the cleavage site, while in these two proteins, they're AT-rich, and they're distal to the cleavage site. So we felt that there was an opportunity to study these new Cas proteins with very different properties from canonical Cas9. So I'll focus on C2C1, the second of these two proteins, where Hui Yang in my lab was able to generate a complex of C2C1 with an RNA that contains both the CRISPR RNA and the transactivating RNA covalently linked as a binary complex, and then also as a ternary complex with a DNA where we have the PAM-containing duplex and the target strand that can pair with the complementary strand on the CRISPR RNA. And that structure was solved last year and is shown here. This heteroduplex is in a cavity within the overall scaffold. Here is the PAM duplex. The CRISPR RNA is a magenta. The transactivating RNA is in cyan. So part of the transactivating RNA is involved in protein RNA recognition. Other elements are exposed. So this is a large complex. I want to just focus on a set of features. Most of the protein RNA contacts are non-sequence specific, and I just want to show you one example. So here is the heteroduplex involving recognition between the CRISPR RNA and the target strand of the DNA, and here is the adjacent PAM-containing duplex. And what you see is you don't have stacking along its entire length. There is stacking here, is broken, and then you have stacking here. So why is that? So a portion of this duplex is shown up here, a portion of this duplex is shown up here, down here. And what you see is that you have a break in the continuous stacking because amino acid side chains are stacking on the terminal pairs here and here. In this case, there are glutamines. And finally, the phosphate linking these two stems is targeted by an arginine side chain and a backbone of a glycine. So if we replace this arginine by an alanine, and there's a cleavage assay generating the two strands, what you see is when you replace the arginine by an alanine, the cleavage is attenuated. And when you make the much more disturbing modification, glycine to proline, the cleavage is completely lost. So you get non-specific interactions, but interactions such that you are breaking continuous stacking by stacking of these aromatic side chains on the terminal pairs. On the other hand, the Cas proteins have to recognize the PAM sequence and distinguish between foreign and host. So in the case of C2C1, the PAM sequence is AT rich. It has two AT pairs and a GC pair. So here's one of the AT pairs, here's the second ATP, and here's the third GC. And now what you see is instead of non-specific interactions, the first AT is recognized from both the major and minor groove, the second by fewer interactions, and the terminal GC, as I've shown before, has these glutamine side chains stacking over the terminal pair. So the PAM is the only site where you have these sequence-specific recognitions, and some of the residues, when mutated, result in attenuation of cleavage. Others, like R122, when mutated to alanine, result in total loss of cleavage. So a single amino acid recognizing a base is sufficient when mutated to result in loss of cleavage. Now, I haven't said anything about the DNA being positioned in the catalytic rough C pocket. And here we were very, very lucky. When we generated the ternary complex of C2C1 with RNA and the DNA construct, we added excess of this strand. So the DNA construct contained this strand and this strand. PAM sequence is here. Heteroduplex formation is here. And to our great surprise, we found the excess of this strand threading its way into the catalytic pocket. So here is this excess. It's an 8-mer. We can see residues 3, 4, 5, 6, and 7. The catalytic residues in the rough C pocket are sitting here, and we see an extra sulfate. So here are steps 4 and 5 coming from this excess RNA strand, positioned in the catalytic pocket. In order to form the crystals, we had to mutate all 3 acidic residues to alanine. So we're just modeling the side chains here. There's also a tyrosine, and then an arginine and a serine forming this particular catalytic pocket. Since we know these residues, we can mutate them. In the wild-type protein, if we replace any of these catalytic residues by alanine, cleavage is completely lost. And for the other residues, cleavage is either lost or predominantly lost. So this was very interesting to us. This extra strand was positioned in the catalytic pocket, and there was also a sulfate. So the last 2 base pairs of the heteroduplex are shown here, and here is the 5' end of the target DNA strand, and this extra strand has the 3' end. There's a sulfate in between. So you could possibly link these, and that's exactly what we did. We made another ternary complex where we extended the target strand. And now what you see is here is the heteroduplex, and the extension now is forming a turn and it's positioning the phosphate that has to be cleaved in the catalytic pocket. So here is the end of the duplex, here is the extension, and the extension involves a large, that was my worry. Oops. Involves a large number of hydrogen bonding and stacking interactions, positioning, if somebody could get me an updated pointer, please, positioning the catalytic residues shown in stars opposite the phosphate that has to be cleaved. So that is summarized in this slide. On the upper left is the structure containing the excess strand. On the upper right is the overhang. And what you can see is that they're both positioned in the same way, now positioning a particular phosphate opposite the cleavage site. And when we do biochemical assays, in the lower panel where the triangles are is where the cleavage occurs. And for the target strand, the cleavage that we see biochemically is exactly the cleavage that we see structurally. Oh, thank you. Can you just replace the batteries on that? Sorry, it's pretty weak, but. So the cleavage site is here when we have the extra strand, and the cleavage site is here when we have the overhang. So we said, okay, why don't we try and have an overhang on the non-target strand, shown on the upper right. Now we use the stretch of T's because we didn't want to have the non-target strand pair with the target strand and compete with pairing with the CRISPR strand, CRISPR-RNA strand. And when we solved this structure shown on the upper left, we could see a stretch of T's sitting in the catalytic pocket. But because they were all T's, we couldn't say where the cleavage site was occurring, just say that there is a phosphate that's positioned in the catalytic pocket. We tried to introduce non-T sequences, but unfortunately, the crystals haven't formed. Biochemically, they occur on the non-target strand, where the red arrow is shown. So what is very interesting about this system is that you have one rough C pocket, and it cleaves both the target and the non-target strands. And at this stage, we don't know what the sequential order of cleavage is. I'm really going to need another pointer. This is not working. Now, since we did the binary complex and also the ternary complex, we could compare the two structures. And one interesting feature was the CRISPR-RNA in what we call the seed segment. In the binary complex where there is no DNA, on the left box panel you see residues 1, 2, 3, 4, and 5 on the CRISPR-RNA, and then the rest of the CRISPR-RNA guide is disordered. In the ternary complex, where it's pairing with the DNA, and you have a duplex, you can follow the CRISPR-RNA shown on the right box panel along its full length. But when you look at the figure shown below, in silver is the CRISPR-RNA in the binary complex, and in magenta is the CRISPR-RNA in the ternary complex. And what you see is that a tryptophan residue packs again base 5 in the binary complex, preventing further stacking of the CRISPR-RNA. But in the ternary complex in magenta, the tryptophan moves out of the way, and now you can follow the CRISPR-RNA along its full length. And features like these were also seen in Argonaut proteins. The last comparison I want to make is that we studied not only C2C1 on the left, but also CPF1, the other new nuclease. C2C1 has transactivating RNA, CPF1 lacks it. And the conformational changes that we see from binary to ternary for these two domains, helical 1 and helical 2, are much smaller for C2C1 that has the transactivating RNA than they are for CPF1 that lacks it. So the transactivating RNA makes the overall fold much more compact, resulting in much smaller conformational changes. I really need the pointer. Somebody can help out. It's very weak. I can barely see it. Okay. Now, of course, phages do not want the CRISPR-Cas system to cleave their double-stranded DNA, and so they evolve proteins, anti-CRISPR proteins, that target the CRISPR pathway. And several of these anti-CRISPR proteins have now been identified and can be studied structurally. So now I'm looking at the SPY canonical Cas9, which is targeted by several anti-CRISPR proteins. And I want to focus on one of them, ACR2A4. So what does the anti-CRISPR protein target? Does it target ApoCas9? Does it target the binary complex? Or does it target the ternary complex with bound double-stranded DNA? And in these pull-down experiments, we can show that the inhibitor does not bind ApoCas9, nor does it bind the ternary complex, but instead binds the binary complex containing the RNA. You're able to solve the crystal structure of this inhibitor. It's shown on the left side, both in a ribbon view and in a surface view. Following that, Hui Yang took SP-Cas9, whose domain architecture is shown on top. The RNA contains CRISPR RNA and transactivating RNA, and solved the structure of the binary complex bound by this inhibitor. And in the lower right, you see the inhibitor circle binding to the one surface of Cas9. And this was done both in my lab and in a lab in China. Sorry. That's okay. Even this is weak. Just replace the batteries on the other one. Okay. So what you see is that the inhibitor is binding to a specific site on the complex of Cas9 bound to its RNA. And what is very interesting about this inhibitor is that the binding site of the inhibitor is exactly where the PAM segment of the double-stranded DNA target binds. So basically, the inhibitor is occluding the binding site for recognition of the PAM domain. But it's equally interesting that there's a loop on the inhibitor which projects out and enters the catalytic pocket. So what you're seeing here is this loop segment entering and positioning itself opposite the catalytic residues. So the inhibitor seems to have two functions. It occludes the PAM site and also inserts a loop into the catalytic pocket. So thus preventing both double-stranded DNA binding, and even if the double-stranded DNA bound, it would not have access to the catalytic pocket. Does the binding of the inhibitor result in a conformational change in the Cas9? When we go from the RNA-bound binary complex and add double-stranded DNA, there are very large conformational changes in Cas9. But if you add the inhibitor to the binary complex, there are very small, minor changes, suggesting that the architecture of the RNA-bound Cas9 is locked in place and the inhibitor binds it without additional conformational changes. Where is this field moving? What is the exciting challenge in the future? There was a third Cas that was identified called C2C2 in Feng Zheng's lab, and later on in the Doudna lab, it was shown that it is not only involved in target cleavage, but also in precursor to mature CRISPR RNA processing. This enzyme has a totally different architecture. It has these HEPN domains, a pair of them. They are basic arginine and histidine-rich catalytic domains. And what is very interesting about C2C2 is that does not cleave double-stranded DNA, but it cleaves RNA. So now we have a Cas nuclease that cleaves RNA, and it cleaves it in a promiscuous manner. So the cleavage sites can occur in single-stranded regions and in stem loops. Oops, sorry. So Yan Li, who was a postdoc in my lab, who did all the Argonaut work in the lab, has her own lab at the Institute of Biophysics in Beijing. And this year, she reported on the structure of C2C2 bound to mature CRISPR RNA. And as you can see, the architecture is extremely different from canonical Cas proteins. As far as the RNA in the complex is concerned, they can see the stem loop repeat, but the spacer, only a small segment of the spacer can be observed. Most of it is disordered in the binary complex. And finally, the two HEPN domains that are separated in the secondary fold come together in the tertiary fold with one set of catalytic residues here and a second set of catalytic residues here. So the challenge now is to make the ternary complexes with target RNAs, see if they pair with the CRISPR RNA spacer, and what are the overall conformational changes that will occur as one goes from the binary to the ternary complex. Now, this work was done by two very talented postdocs, Aiming Ren, who worked on riboswitches and ribozymes, and has her own lab now back in China. And the CRISPR-Cas work was done by Hui Yang, who's currently in the lab. Now, I want to end by very briefly presenting ongoing work with the lab of Sriram Subramaniam at the NCI. Now, I've talked about Cas proteins as effectors, single domain proteins, but here I'm talking about cascade, another effector which involves a multi-subunit component. So here I'm showing you a type 1F cascade containing a spine of 7.6 subunits, plus a 6F subunit, a 5F subunit, and an 8F subunit. And here is the CRISPR RNA with its seed segment, a 5' end, with a stem loop at its 3' end. So the protein is much larger. It's amenable to study by both crystallography and cryoEM. And we are interested in anti-CRISPR proteins that target cascade. Our crystallographic efforts didn't work, and so we turned with Sriram to work on the cryoEM structures. And to date, we've looked at 3 anti-CRISPR proteins called 1, 2, and 10. For 1 and 10, we have the X-ray structures of the anti-CRISPR proteins. And working with Sriram's lab, we have solved in this particular view the structure of cascade bound to 2 ACRF1 anti-CRISPR proteins. And what you see is that the anti-CRISPR proteins are targeting the binary complex of cascade with the CRISPR RNA at sites where the CRISPR RNA would pair with the target DNA. So basically, ACRF1 with these 2 bound ACRF1s is occluding binding of the double-stranded DNA and therefore preventing cleavage of the invading phage double-stranded DNA. What about 2 and 10? Here is ACRF2 binding here. Here is ACRF10 binding here. So 2 and 10 are binding at sites where the PAM segment of the double-stranded DNA would be positioned on the cascade scaffold. So both inhibitors are targeting the same site, but because these are different proteins, they are doing so in a different manner. So So overall, here's the cascade scaffold bound with CRISPR-RNA, shown in cyan. And ACRF1, 2 molecules, are targeting a phase involving recognition of the spacer element of the RNA with the double-stranded DNA. So they're occluding binding of DNA, and ACRF2 and 10 are occluding binding of the PAM site on the DNA. There are more inhibitors in this family, and these have to be studied to see if they bind by common principles or using different principles. And I asked Sriram for a slide of his lab. We have competition in this area, so Sriram has put a lot of members of his team on the project, and Taiwei is the one, a pre-MD student in his lab that we have our most interactions with. So I'll stop here. I hope I've given you an idea using riboswitches, a feeling for RNA architecture, for RNA pockets, how ligands are recognized in these pockets and discriminated against, about conformational transitions when ligands bind. And then in the case of ribozymes, small RNAs that contain specific sites for cleavage, where we can look at the architecture of those sites, look for splayed apart alignments for bases that could be involved in activation and stabilization of the cleavage step. And finally, in the CRISPR-Cas field, we have worked with these new nucleases that have appeared in the last year or so that involve a staggered cleavage, a single nuclease pocket, and studied both their binary and ternary complexes and learned something about how the catalytic pockets function, how the cleavage sites occur, and so on. And finally, we are interested in anti-CRISPR proteins that target both cascade as well as Cas, and we have used both X-ray crystallography and cryoEM to address these issues and try and understand how these anti-CRISPR proteins function. Do they occlude DNA at the heteroduplex level, or do they occlude recognition of the PAM sequence, or do they occlude access to the catalytic pocket? Thank you very much.
There's time for 1 or 2 questions, else we move to the library, and you can discuss with Dinshal there.
Thank you. Very elegant studies. My mind is usually on the application of all these structural and mechanism of RNA or DNA movement or architecture. Have you noticed any changes in energy transfer when you substitute one molecule? For example, if you substitute glutamine for arginine and or another charged molecule which is occupying a space, and it has to be somewhat energy involved. Plus, have you noticed, I mean, maybe it's not fair to ask you, but have you noticed when you have conformational and deconformational status of the RNA, that there is a change in energy dynamics?
So let me try and answer your question the following way. The glutamine riboswitch is very specific for glutamine. You cannot replace it by any other amino acid. You can't even replace it by L-glutamate. So one interesting question is, you have the structure of the glutamine riboswitch. Can you make changes either in the pocket or far from the pocket so that the glutamine riboswitch would bind glutamate, which has a charge? And that is not straightforward. because work we've done earlier tells us that the changes that occur not only occur in the catalytic pocket or binding pocket, but also far away from the binding pocket. So we looked at a purine riboswitch with and without a sugar moiety attached to it. And the changes were not only in the catalytic pocket, but they were also far from the catalytic pocket.
You assume, right?
So it's very difficult to try and take a structure and remodel the pocket to bind a close analog. It's very difficult.
What about in the presence of your the inhibitors?
You're talking of the anti-CRISPR?
Right. Some of the inhibitors you mentioned.
So that work is very new.
Okay.
And since they are proteins, we could look at the interface and replace amino acid changes so as to change the electrostatics.
Right.
See what the consequences are. And it's all very new, and we haven't done anything along those lines.
Great. Thank you.
So let's thank Dinsha again and meet in the library.